This video outlines the MacConkey medium. MacConkey medium is considered a selective and differential medium. It is selective because it has two ingredients, crystal violet and bile salts, that both interfere with formation of the peptidoglycan layer in gram-positive cells. So therefore we can say it selects against gram-positive cells, so they do not grow on this medium. So if the organism is growing in the medium, we can assume it's a gram-negative bacterium. So that's the selective aspect. The differential portion of the medium is due to the presence of the disaccharide lactose. If the organism can ferment lactose, it interacts with a pH indicator because when the fermentation of lactose occurs, the pH drops below 7. And the pH indicator in the medium is called neutral red. And that neutral red will turn a bright pink color. So the organism first must be able to grow, and then we can say it's a gram-negative, and then we can determine whether it is an organism that has the enzyme lactase, which allows it to ferment lactose. Pay very close attention to those endings when you're describing the medium. Okay, so let's lift this up so we can get a little bit better look. Here we go. Okay, so here you can see at the very top is not only an organism that can grow on this medium, so now we know it's a gram-negative, but it can also ferment the disaccharide we talked about, lactose. And that means this particular organism has the lactase enzyme. So let's compare that to an organism that grows but doesn't have that appearance. And this is our differential aspect here. So now we have an organism that does grow, so it is a gram-negative, but you can see it is clear. You don't see that color change going out into the medium. Now keep in mind that the medium itself has a bit of a tint to it because it's got crystal violet and neutral red, so don't confuse that sort of background color on the medium with the ability to ferment lactose. So here you see no color change out into the medium. It's certainly not that bright pink color. So we say that this organism lacks or doesn't have the enzyme lactase. It is unable to ferment lactose, as opposed to the one we looked at previously with that bright pink color that does have the enzyme lactase and can ferment lactose. Okay, so let's look at a third possibility when we put an organism on this particular medium. And in this area we see no growth. So compare that to the growth that we actually saw there with no fermentation, so you need to look very carefully. This would seem to indicate that the organism is gram-positive, and so therefore the crystal violet and bile salts interfere with its peptidoglycan layer and it's unable to grow. We can't really make any determination on its ability to ferment lactose because it didn't grow on this medium. Okay, so in review, again, MacConkey's is a selective and differential medium. It is selective because of the ingredients crystal violet and bile salts, which prevent gram-positive bacteria from growing on the medium. And so we can say that it selects against gram-positive bacteria, or we can also think of it as it selects for gram-negatives, allowing them to grow. So we also said that this is differential as well. And so because there is a sugar in there, lactose, we can look to see if the organism can ferment or break down that sugar with the enzyme lactase. If the organism can ferment or break down lactose, we get this nice bright pink color on the medium, and that is due to a pH indicator called neutral red. So as the pH drops, the neutral red produces that nice bright pink color. And we say that these organisms are coliforms that can ferment lactose, and we generally put them in the non-pathogen category. Compared to an organism that can grow, but yet cannot ferment lactose, we call these organisms non-coliforms, and we say that they may be pathogenic. So these types of organisms lack the enzyme lactase. And then finally, if you put a gram-positive organism on here, we shouldn't see any growth in that area. And that is an overview of the MacConkey medium.